just want to open by thanking all the organizers for bringing us all together. I'm really looking forward to looking at the posters that they can see in the back of the room at the break. Touch is our most intimate sense. On a clear day, we can see for miles. With the right acoustics or technology, we can speak and be heard at the back of a large room or even across hundreds of thousands of miles. But touch demands contact. It does not work at a distance. So I want you to spend the next 20 minutes or so thinking about what you would need to know to be able to decipher how touch happens. And I'm going to tell you about what my team of biologists and engineers and chemists and physicists and computer scientists have been doing to address that problem over the last few years. Much of this involves developing tools to deliver mechanical stimuli in controlled fashion to measure the impact of forces in biological systems. And I promise you there will be an ion channel, so don't fear. We're going to get there. And also molecular tools to visualize how things are happening. So touch is a really complicated sense. It has many different elements to it. We have gentle touch, we have effective touch, we have touch that feels painful, we have a variety of sensory neurons that are embedded in our skin that mediate those perceptions. I like things simple. So we've been working with the nematode coelians because it provides a variety of ways for us to go ahead and investigate touch sensation. So what you're seeing here is a transgenic animal expressing GFP in every single one of the 302 neurons that are present in C. elegans. They all have names. They're the same from animal to animal, and 6 of them are so-called gentle touch receptor neurons. A pair in the tail, a pair in the head, and 2 neurons that innervate the spectral surface. When you see pictures of worms in my talk, the round end is the head. That would be on this side of the slide. The pointy end is the tail. That would be on that side of the slide. So it was known that these 6 touch receptor neurons were required for behavioral responses to touch, but it wasn't entirely clear what their range of sensitivity was. So working with Beth Pruitt's group and students that were co-advised by Beth and myself, we developed new technology that allowed us to deliver forces from a calibrated cantilever. And then we can measure the response probability as a function of the force that was delivered to the cantilever. So Eileen Mazzucchetti and Adam Kempkin did this work, and what we find from this is that we saturate the response with about 1 micronewtons of force. So for comparison, if we were to do a test on the average human palm, you would be able to detect 1 micronewton. You need about 10, okay? So these animals are highly sensitive to touch, which is another feature in addition to there only being 6 touch receptor neurons that makes them useful. In addition, genetic studies have already identified a series of genes, including this one here, that when mutates, essentially suppress the response to touch. What does this gene encode? I promised you ion channels. It encodes an ion channel. So MEC4, together with the subunits of alpha, beta, and gamma ENaC, was one of the first ion channels identified in the large family of ENaC channels called degenerate ENaCs and ASICs. An interesting little tidbit about this class of ion channels is that they are only present in animal genomes. You will not find them in any other kind of organism, which is kind of cool. So in order to actually understand how MEC4 worked as an ion channel in the touch receptor neurons to make those behavioral responses possible, we worked out how to record from identified sensory neurons in a living animal and to deliver a mechanical stimulus, which is shown in this trace here. And then what we see here now is a mechanical receptor potential. So there are a couple of things about this that were surprising at the time that we first collected the data, which is that the channel is activated at the onset and then the removal of the stimulus. Both the onset and the removal responses require the MEC4 gene. And in other data I don't have time to show you, we also showed that we could modify this channel, this gene, and then also modify the selectivity of the channel. So this is really the channel that's doing the business. So we also wanted to know where is this channel in the touch receptor neurons. So we built on antibodies that were specific for the touch receptor neuron, and we can see that it localizes to these discrete puncta. Fast forward several years, and I can generate a guess at what the structure of these channels might be based on both crystalline cryo-ion structures of a related channel called ASIC1A. So this is not MEC4, but this is what we imagine it might look like based on homology. So the channels form trimers. They have a large intracellular domain and a modest intercellular domain. So I mentioned that this onset and offset was surprising. And there's some other characteristics about the channel properties I'll show. So they're collective as we'd expect for an ENaC-like channel, and they're blocked by amiloride, again, also a shared feature. While these response dynamics are not exclusive to the touch receptor neurons, they're also found in nociceptors in C. elegans and in texture sensors in C. elegans and in another class of nociceptors in C. elegans. So some of these studies are from my group, some of them are from other groups. That also makes me super happy that this is something that other people are finding. And quite recently, in fact, we also now see that these response dynamics are also seen in a vertebrate mechanoreceptor, okay? So one thing that I want to point out, and I'll slide back to this slide for a moment, is that each one of these currents that you're seeing here that have similar response dynamics, they're carried by different ion channels, okay? And that's probably also true of these data, these data from Slav Defencio's lab in the duck, okay? So these response dynamics are not uniquely identified with a particular kind of ion channel. And one thing that tells me as a biophysicist is that the response dynamics reflect conserved biophysics of how force gets from the skin to the channels in the sensory neurons. And nature has grabbed whatever ion channels happen to be available to hook up to that apparatus to make that work. Okay. So I really love this sculpture because the artist understood something and represented it here in marble, that when the skin is indented in one location, it is deformed at a distance from that. So when we're thinking about how touch happens, we also need to think about where it happens, okay, and the mechanics of how that deformation spreads from the point of indentation. So to look at that in our favorite C. elegans model system, we built a microfluidics chamber that held worms in place and then could deliver a mechanical stimulus while we were observing it. And in order to, so our hypothesis was that when we push on the skin, we would deform the skin, which had been demonstrated by other groups, and that deformation of the skin would also be translated to a local change in strain in the sensory neuron, which extends along the length of the body, as we saw a few slides ago. So in order to see that, we kind of needed fiducial markers. And what we did was we took advantage of the fact that these neurons have very large mitochondria. So these are mitochondria tied with GFP. They're very stable, they don't move. So we can use them as fiducial markers to see what happens when we push on the skin. And what we found was that when we deliver that stimulus here, we would see a local increase in strain that we were reading out by the relative motion movement of those mitochondria. Okay, so now let's kind of try to put all of these observations together into a biophysical model about how force might be getting from the skin to these ion channels. So we developed this model in collaboration with Massimo Vergassola, and it's built on classical models of indentation of a cylinder here, shown with a nice little video. And the idea is that when we indent, we deform, and then there's a restoring force because of the shape of the tissue that pushes back at the end to account for the activation of the onset and the offset. And you can write this out in math that runs something like this. And if anyone wants to get into this deeper, I'm happy to do that at the break. So what does it look like when we do that kind of an indentation dynamically in our nematodes? So again, a new tool. So this allows us to deliver rapid indentations. And we have always, this is in 20 micron. So you can see that each time we're pushing into the animal a little bit deeper. And when we do that, currents get bigger as we push deeper. So that could indicate that when we're pushing deeper, we're increasing the probability that the channels that are affected by that local strain are opening. But it could also mean that as we're pushing deeper, we're also stretching further away from where the indentation is being delivered. And what we see when we just look at the current as a function of displacement, it keeps going up and it doesn't really saturate. And then we built a simulation that connected that indentation to activation of channels. And in the simulation, we could separate the change in open probability from the likelihood of recruiting channels at a distance. So in the simulation, the change in open probability changes within less than 1 micron, that smallest indentation. And then what happens as we start to increase the displacement is that we're recruiting more and more channels at a distance. Okay. A consequence of the mechanical model is that when we change the speed at which we do the indentation, we're also changing the extent of the indentation. So slower indentations are going to have a smaller effect on deformation at a distance, and faster ones are going to have a bigger effect. So with our stimulator, we can also regulate the speed. So if the slowest speed's up here at the onset, pretty much the channels don't open. They're insensitive to slow stimuli. And as we increase the speed, we get larger and larger currents. And this is quantified over here on the right and then simulated down. So all of this together suggests, and I already showed you, that the MEC4 channels are located in discrete puncta. It also suggests that those discrete puncta are probably not very mobile, because it won't really be very useful to recruit channels at a distance if they're continuously moving around. So we had noticed in some of our EM work earlier, so what I'm showing you here, this is a cross-section of a touch receptor neuron. It has many, many microtubules. Those are not synaptic vesicles, so if you're used to looking at synapses, that might look like that to you. And then the neuron itself is embedded inside a skin cell. And in between, there are these really curious electron-dense structures, which we can reconstruct in 3 dimensions over here on the right. So one of the things we wanted to know, so our initial TEMs were about 3 micron segments, and you can see we have these clanks or beams. They're about a micron long, but we couldn't tell how they were distributed from these short segments. So we teamed up with Mazen in Toronto, who had already collected large zero-section data sets for connectomics, and then we looked at them to look for these structures. And what we found is that there were segments of the neuron that had none, some, none, some, and like this, and that their distribution, on average, about 1.5 microns end to end, and they're about a micron long. Well, that turns out to match what we see when we look at four next here, not tagged with an antibody, but rather tagged endogenously within neon green fluorescence that we see responded. So what might hold it in place? So Alka Das, when she was in the lab, obtained or generated a set of 20 strains, endogenously tagging conserved extracellular matrix proteins, and we found two. This distribution was very similar to MEC4's. All right, is that just a coincidence? Let's go find out. Sure. First, I want to tell you, not only is MEC4 in these discrete puncta, but they don't move. So this is a chronogram of an image of the same neuron collected over 30 minutes. Pretty straight lines, right? So we've got time on this dimension and the space on this dimension. We bleached this one, and it doesn't recover visibly during that time frame, nor do its neighbors dim. We can quantify that with a shorter-term FAP experiment, which I'm showing you here, so MEC4, where we see a very modest recovery over that 3-minute time period. And this is not something that's just weird about this neuron, that all membrane proteins are immobile, because we can look at another membrane protein, Sophion, which is also expressed in the touch receptor neurons, and you see much greater recovery in that FAP experiments. All right, so what about those two ECM proteins? So they are also similarly stable and immobile along the touch receptor neurons. Now, these are highly conserved essential elements of basement membranes throughout the animal. Another group led by David Sherwood, who first created these two lines that I'm showing you here, did FAP experiments in another tissue and showed in other tissue, for example, for nitigen, you would see about a 30% recovery in this 5-minute FAP experiment. So there's something different about this environment, this intersection between a neuron and a skin cell than the basement membrane elsewhere in the same animal with the same marker. So we have similar immobility, and we also have similar distribution. So what I'm showing you here are basically the statistics of the interpunctum intervals for laminin, nitigen, and our MEC4 channel. All right, so circumstantial evidence that they're connected. Now let's get more detailed. So we can generate an animal that labels both the channel and one of these proteins in different colors. And then we can ask questions about co-localization, which we have done so far in the ventral line film microscopy. Super resolution work is ongoing. So let's walk our way through this. So here we have an animal that labels NID1 in 2 colors. Okay, NID1 forms complexes with laminin and with itself, so this is kind of a positive control for association between 2 proteins. And at the bottom, we have MEC4 and another membrane protein in the touch receptor neuron, and there's no particular reason to think that they'll be adjusted. So over here on the right is a quantification of the correlation in intensity, some intensity correlation quotient. Perfect correlation is out here at 0.5. No correlation is down here at 0. So for our controls, basically no correlation, and then out here we have a correlation. Now I'm going to tell you about what this box is. So these are paired images analyzed, and then here we shuffle the images, and we took images from different animals in the 2 colors, and we made the same calculation. So in that case, if this is a real association, then we ought to see very little correlation when we have these shuffled pairs, and that is what we'll see. So now for the real meat of things, we have nitigen 1, NID1, and MEC4. Their intensity is highly correlated and not when we shuffle, and similarly for one of the laminin subunits, together with MEC4, we see correlation that's distinct in the actual pairs versus the shuffled pairs. Unfortunately, we can't ask what happens when animals are missing laminin because they don't survive, but we can ask what happens when they're missing nivogen. This image is maybe a little bit difficult to see, but this is an animal that lacks the nivogen protein, and maybe from these intensity traces down below, it's a little bit easier to see that, yeah, there are some puncta which are correlated and some which are not. And then when I look at the distribution of both MEC4 and laminin in these median mutants, we see that there's an increase in spacing between the adjacent puncta, that is, they become less steps. So the gray hairs are wild type and the colored traces are the mutants for these two proteins in these two conditions. So what happens functionally? So we can perform a behavioral test of touch sensation, and we see that there's a modest decrease in sensitivity in the NIV1 mutants. And for comparison, I'm showing below an animal that lacks the MEC4 ion channel and an animal that lacks a different extracellular matrix protein that was previously identified as being required for touch sensation. And in the center, you'll see electrophysiological recordings from those three genotypes. And then a summary of the size of the current as a function of mutation. So it's control, NIV1, and MEC5 at the bottom. So there's basically no current in MEC5 at any intensity. But in NIV1, there are some complexes that are associated with one another that continue to function, but there are fewer of them. So if you're thinking about what I showed you earlier, then that kind of makes sense in terms of the localization. All right, so in the last 3 minutes or so, I'm going to tell you about one previously identified extracellular matrix protein. So it was known to be required for touch sensation. All three of these, MEC1, MEC5, and MEC9, known to be required for touch sensation and needed for the distribution of MEC4 channels. I want to dive into MEC1 in particular. So MEC1 encodes a very large secreted protein, and it has in it 15 predicted kunits or protease inhibitor domains. And those of you who are knowledgeable about ion channel toxins will recognize that this fold is the same as the fold that's found in cysteine knot peptide toxins of ion channels. So what I'm showing you down below here is an alignment between the 15th kunits domain in MEC1 and a toxin that's known to bind to ASIC1A, which is in the same superfamily of the degenerate ENAC site I mentioned earlier. So we were curious. So we know that when we get rid of MEC1, MEC4 doesn't stick in these puncta. NIV1 doesn't stick in these puncta. I didn't show you those data for time considerations, but that's what we know. What happens if we manipulate this terminal kunits domain and some of the conserved cysteines that are required for folding that domain? So we did that in a couple of ways. So we took an existing mutation that came out of the forward genetic screen, so we already knew that that mutation caused behavioral defects, and that's the arginine to cysteine mutation. And that has a minor effect on the distribution of NIV1. Then we also mutated a nearby cysteine that's one of the ones that's required for folding in these domains. And we did 2 mutations, and we see a bigger effect in terms of reduced distribution. But what about function and association with MEC4 channel? All 3 of those mutations reduce the correlation in intensity between MEC4 and NIV1, and they have dramatic effects on touch sensation. So now I can kind of pull all of these things together for you into a model which is comprehensive in the sense that we can think about what happens from the first steps in touch, the contact between the stimulation and the skin and the ion channels in the sensory neurons that are activated and that give rise to behavioral responses to touch and our perceptions of time. So we have, okay, so up here, we're indenting the skin and we're stretching the neuron locally, increasing strain, as I showed you. We imagine that that increase in strain is going to pull on those ECM beams, and that those ECM beams have as part of their constituents mitogen and laminin, which I've mentioned in the talk so far, and that MEC1 being such this large protein, and knowing that we can basically disrupt the C-terminal end of it and disrupt function. I didn't show you, but that maintains the distribution of the ECM proteins, but disrupts MEC4, which is critical for this model. So here, we can't really understand how this mechano-sensitive channel works in isolation, because it works in a system and in the tissue in which the neurons are closely connected to skin cells. And in fact, MEC1 and methionine, which I'm going to talk about, they're made by the neuron, right? And mitogen and laminin is made by the epidermal cell. So we have protein products that are secreted into the space in between these two cell types that are required for this ion channel to function as a mechanosensitive channel. So is this interface unique to the worms? No. There are analogous interfaces between sensory neurons that have been documented in Drosophila and zebrafish, and even in human skin. In the outermost layers of your skin, there are unmyelinated nerve fibers that literally travel through tunnels in the skin cells. So this close association between our nerves and our skin cells seems to be something that is found across many animals. So we have conservation of biophysical responses to mechanical stimuli, and also how tissues are built to make touch happen. So I've talked about the work that many of the people in the lab, and those highlighted in yellow are members of the lab currently. I've mentioned several of our collaborators, and I also want to express my gratitude to the American taxpayer and to NINDS for funding this work. Thank you.
Great, so cool. And my question is, given the setup of this system and the way the toxin domains bind things together, but what do you think is that fundamentally sets the threshold of sensitivity, you were saying around 1 microdude? Is it I guess kind of conceptually, do you think it's a physical limitation of the system and you can't go below 1 micronewton, or that below 1 micronewton might just be physiologically irrelevant? Or and kind of bet in that is, how far could you in theory push this kind of system to fuck up that area expansion needs to make a force sensitive?
Okay, so let me try to unpack that. So I think what you're asking about is like what regulates the sensitivity, okay, to touch kind of go when we think of ways in which activity modifier. So there's some things I didn't have time to tell you. One of which is that timing matters. So I showed you what happens when you move slowly versus quickly. I didn't show you what happens if we give a sinusoidal stimulus. These neurons and classes of neurons that we use to protect our cell phones by living in our pockets, they're quite selectively responsive to vibration. So when you have a vibration stimulus, right, that actually is more efficiently transmitted through the tissue, okay, and generating responses. neurons, and then also get repeated responses. So some of the sensitivity also, there's an interaction with frequencies of activity. Okay. And for the worm, we see maximal sensitivity about 1 kilohertz. And that's actually a little bit faster. That's what the biggest in humans, but there's a wide range. That's like 500 hertz-ish sensitivity. Okay. So some of it has to do with the temporal dynamics of how forces transcend from the stem to the program, right? And the closer the neuron is to the stem, which is the case here, right? The more closely coupled it's going to be to us. Thanks.
I like a peer guy, but I did have a question. So I was really curious about the elasticity model or the skin model. And with aging, right, you lose touch sensation, and that's often attributed to loss, perhaps, of innervation of the skin. But I was wondering if your model might capture how differences in skin elasticity could affect recruitment of ion channels along nerves in the skin, and if you have looked at maybe age ones, or if you plan to, and again, if like your the nice modeling data that you had there, that have to capture that to understand how much of it might be loss of innervation versus changes in elasticity or some kind of synergistic effect of the two.
Great questions. So we have a bunch of listed that ask those questions, and Sophion is a great model for agings that have a very short life, about 2 weeks on average. So we haven't yet begin to look at that explicitly with all of my physical tools, but I can tell you from a behavioral standpoint, animals as they reach forms as they age, you have, I think, these mechano-sensitivity. A key question might be, is that coupled with the change with the distribution of the channels and ECM structures? Because we put that yet, we haven't looked at the history of point atoms. And then in terms of modeling, we can also look at some elements of the mechanics of the skin, which we have some early data about to see how that is. So in humans, right, there's a lot of complex things going on. Another piece to think about, what are the communications between neurons and the skin cells? So our skin turns over all the time. We're generating new skin. As we get older, that process is less effective. Takes longer to generate a piece of skin. Our neurons don't turn over, make new cells in the same way that our skin does. So we have a tissue where we have skin cells that are very dependent and neurons which aren't, but yet we want to have a stable sensory perception. So I think that's also a really interesting question into the future. Thanks for the question. Okay.
Hey, Gio.